Alright, section 5.5 simply deals with objective number 9 here. Name 2 non-cellular infectious agents besides viruses and what they infect specifically. So the first one are prions. And prions are interesting molecules. They are composed completely of protein. That's it. They don't have carbohydrates with them. They have no nucleic acid. So you might wonder, how is it propagated? How is it multiplied? And there's still questions that remain. Doctors and scientists still do not fully understand how prions work. They have a basic understanding of it, but the nitty-gritty of it, how to stop it once a person has a prion or an animal has a prion in them, they don't understand how to keep it from propagating. Alright, so you don't have very many pictures, if any, in your textbook that look at prions. And it's interesting. You can probably find videos on prions on YouTube or on Google. And it's interesting because you actually have a prion protein, a protein called PRPC in your brain. This is a neurological protein found in the brain. And the green here is the normal protein, all right, PRPC. And green is green for go. If you have this, everyone does, this normal PRPC protein, things are good. However, if you contract this prion protein, which is depicted as PRPC-SC, or if you contract just the PRPSC, the mutant one, or if your wild type, the normal PRPC protein, somehow randomly mutates, right, to convert to the damaging PRPSC protein. All right. Once this is in your body, what it does, and this is the part that is unknown, it will actually dimerize with the wild type. And so here you have a green and a red together. And over time, it actually converts the normal PRPC protein to the PRPC-SC. And then you have all PRPSC proteins. And that is when you start to see neurological dysfunction. All right, so that previous picture and then this table is taken from Wikipedia, which I generally don't like to use, but it was a very basic figure and actually a pretty good diagram of what happens. And then this is a true table of the different diseases that are caused by prions and the different animals that are affected. So some of these you have probably heard of before. In sheep and goat, you can get scrapies. In cattle, you can get bovine spongiform encephalitis, BSE, and a different disease, mad cow disease, and probably the mad cow disease is what people have seen. There are videos of cows that are in this disease condition, and if you see it, you know, in some cases your first response might be to chuckle a little bit, because with the neurological damage, the animal just lacks all cognitive function, or, you know, cognitive makes it seem like they're intelligent, but if you imagine a large four-legged animal on a sheet of ice, and how their feet, you know, in cartoons, they kind of dance on the grass, on the ice before their legs fall out from under them and then they fall down. All right, you can find videos on YouTube, on the internet, that depict this, even though there's no ice underneath the cow. And that's because their brain, their neurological system is just completely racked of all function. And it's not a humane way to die, and it's just awful. Other animals are affected. There's the mink, and this is an encephalopathy as well. And the encephalopathy is just indicating its brain. There's pathology that's going on in the brain, and basically you just create these holes, or holes are created by the presence of the prion proteins. In deer and moose and elk, you can have what's called chronic wasting disease, CWD. This is where one of the manifestations of this disease is that the animals just simply don't eat anymore. And so they waste away. And it's chronic, it's long-term. And you can imagine these very large animals, when they're healthy, it will take time for them to waste away and die from malnourishment. Basically starve to death because of the mass that they start with. Alright, other animals can have this or develop this encephalopathy. It's interesting that the ostrich can be affected as well. But then looking down to humans, there are quite a few different diseases. There's this Creutzfeldt-Jakob disease, and then different varieties of that. And then Kuru, that's probably the most well-known disease, and this was identified in the New Guinea population, where individuals or cultural activity would be to ingest the brain of basically the family patriarch, the male of the family, and that was part of their culture and a very important cultural ritual when it comes to a death in the family. But unfortunately, if these individuals had Kuru and they ingested that brain, which would have the infectious prion protein, then those individuals would then consume that infectious protein, and it wouldn't be too long before they would start to show signs of the Kuru disease, right? Basically the mad cow disease, but only in humans. And it took a while for scientists with the World Health Organization to figure this out. And then, as you can imagine, it also took time for these strangers, the WHO individuals, to be able to create an understanding in these native tribes and cultures that this process of honoring their death, their deceased family members, was something that they needed to change, and that by consuming the brain, they were just actually allowing this process, this disease, to continue. And once they were able to educate the population, this disease isn't seen very often, or Kuru isn't seen very often. Now, there are some other types of encephalopathy that are due to prion diseases, and again, for an unknown reason, these prion proteins can randomly mutate, and so then that's why you have the presence of these diseases. The nice thing is that these are not transmitted from person to person. Just by contact, you actually have to ingest the material. They can be transmitted by blood transfusion in some cases, and so that is why there are history questionnaires when you give blood to see if you've been in different areas of the world where mad cow disease may have been present. Because if you've eaten, say, meat from a cow who had mad cow disease, then there could be the chance of you contracting the prion protein. Oh, sorry. So, that is prions. Very interesting, but very difficult to understand. All right, the last one is viroids. Viroids are virus-like agents. All right, so you got the V and the V, virus and viroids, but viroids are only infectious to plants. So they parasitize plants. They are much smaller than viruses, and it's interesting because they simply contain a strand of RNA. All right, they don't have the capsid coat. They don't have any other type of coating, no protein, just the RNA. And they are significant pathogens to crops. All right, tomatoes, potatoes, cucumbers, citrus trees, chrysanthemums. So many plants are at risk of contracting these, but the nice thing is that they are actually specific to the plant. So a viroid that targets tomatoes won't cross over and target potatoes. Right, but viroids, virus-like agents that parasitize plants, and that is what you need to know. All right, then just some review questions. Antibiotics are an effective method for treating viral infections. Cue Jeopardy sound, song, and the answer is false. Antibiotics are not effective against viral infections. All right, antibiotics are going to target the growing and replicating bacteria, whereas viruses are non-living. Right? They are active, but not alive, and they don't have any metabolic processes that a medication can target or an antibiotic can target. There are medications that will be antiviral in nature, but those medications are not classified as antibiotics. All right, then put the phases of the life cycle of animal viruses in the correct order. So this was from section 5.3, I believe, looking at the life cycle. So your options are assembly, penetration, release, absorption, synthesis, and uncoding. And the proper order is adsorption, where the virus attaches to a host cell. Step 2 is where it's going to penetrate to get into the host cell, and then it's going to uncoat. And in your figure, penetration and uncoating were part of the same step. Then there's going to be synthesis, where the host cell is going to replicate the nucleic acid for the virus as well as create the capsids. Then there is the assembly of the virion, and then release. And then the virion can move out and infect another host cell. All right, that is it for section 5.5, looking at prions and viroids and a short review of some of the other topics.